Hi, I'm Dr. Greg Nigh, and welcome back to the deep dive. Today, I'm taking on kind of a complex topic, but I'm just gonna sort of skim the surface on it. I want to talk about methylation, which there's a whole industry now about MTHFR and methylation, and everybody, patients come in, they tell me I can't methylate, and I've got MTHFR. And the other biggie is with SIBO, that people are, come in with tests that they are testing positive for methane SIBO. And the standard approach in with SIBO, as I talked about in a previous video, so hopefully I can point, my web guy will put it there. Um, the standard approach to SIBO is like, well, you find out what gases are being produced, with the breath test. You say, oh yeah, it's hydrogen and methane, hydrogen sulfide, and then the strategy very commonly is to kill the bugs, kill the bacteria that are generating those gases, with the hope of long-term relief from SIBO. Well, I have this whack idea that bodies do things because they're trying to fix problems. I mean, we know this, at a certain level, inflammation around a sprained ankle. We know that the inflammation is there because it's trying to fix the problem of the sprained ankle. But then, and then there are other examples. I mean, they abound of how what are called symptoms are actually maybe, like a fever. Well, we need to suppress that. No, maybe we need to let that go because the body needs to enhance metabolism to take care of the problem. So, SIBO has come to be defined as one of these gases or more being produced in excess and making people miserable. So in a previous podcast, I kind of gave an overview of these gases that are being produced and teased the idea that, maybe this is happening to try to fix a problem. So today, fairly briefly, because it's obviously a very deep biochemical topic, but I want to just talk about methane gas and sort of reframe what's going on with methane, which it's very hard to find in sort of in the popular discussion about methane and excess methane and methane SIBO, anybody saying what methane is good for. Like maybe if we look at what it's good for, we could then work backwards and try to figure out why would the body be making more of it. So I am once again gonna try to share my screen and let's see how this works. Maybe I'll do it this way. Let's see. Do I got it? Yes. Okay. So this is this is I mean this is from a paper that we have recently submitted for publication. It's now, I think it's got accepted. I'm not exactly sure about this one, but it's in the process. Um, so generally, the methyl cycle, the methylation cycle, kind of the image that many of us have in our head because we've stared at it for a long time, is you have methane coming in either dietarily or by gut bacteria. Methane, and it goes around, doesn't really go around, but there's a series of reactions that generate a S-adenosyl methane, SAMe, spits off one of those groups. And as it works its way around the cycle, it gets to homocysteine. And then from homocysteine, it either goes back up to methane via BHMT or via MTHFR, two pathways that it can go, or it goes down the sulfation cycle into the sulfation pathway where it produces glutathione and taurine and ammonia and hydrogen sulfide, all kinds of things down the sulfation pathway. and it just keeps spinning. And many people, various testing or whatever, they find out, oh, I don't methylate very well. And what's that mean? Well, it means methylation is involved in detoxification of all kinds of things, hormones, and methylation is involved in DNA regulation. You got to stick methyl groups on the DNA to suppress gene expression, which is very important around cancer. But our guts produce methane. And methane, if you listen to it, it's got a methyl. There's methyl in there. And our guts are playing a central role in supplying methyl groups to the body. And they're doing it through an astounding series of enzymatic reactions. And what is important to know about the enzyme reactions that I'm going to be pointing to in this complicated pathway in that image, these enzymes have a very high KIE with respect to deuterium. KIE, as I think I explained in the prior video, is the kinetic isotope effect. And if you're not familiar with what I've said about deuterium, you're going to need to watch the other video that kind of gives a deuterium 101 overview. But this is a heavy isotope of hydrogen. It is built into nature. Everywhere you find hydrogen, you're going to find some deuterium, but deuterium is relatively toxic to our cells. So our cells have to be very careful when they're trying to grab a hydrogen that they don't actually grab a deuterium. How do they do that? These enzymes have developed a very strong preference that when they transfer a hydrogen, their choice for hydrogen over deuterium is many, many times greater. So they are very selective so that when they are building new molecules that contain hydrogens, they're very careful that they're not building deuterium into those molecules. Like what kind of molecules? Well, look at this. So we have the depleted hydrogen gas, which again, in the overview I gave of deuterium, I sort of explain where that comes from. And then the gut microbes combine that hydrogen gas with carbon dioxide, and they generate methane, and that converts to methanol, and that converts, oddly enough, to formaldehyde. Formaldehyde, we hear about this chemical that is carcinogenic, and we don't really think of formaldehyde in a healthy context, but formaldehyde is actually a central player in the methylation cycle that's happening in our guts because our bacteria are generating formaldehyde. So, what's very important about formaldehyde is that the hydrogens contained within formaldehyde are depleted of deuterium. Formaldehyde is getting built by the bacteria in a way that the hydrogens are not accidentally deuterium. So there's excess deuterium left over. It's in the surrounding media because if you're being selective for hydrogen, you gotta drop off the deuterium in the environment. And it'll probably be a whole separate video that I'll talk about what becomes of that excess deuterium. So if you look back at this image, we've got formaldehyde now coming in, entering into what we think of as the classic methylation cycle, tetrahydrofolate, which is, precursor to the active form of folate that's used in the body. So tetrahydrofolate combines with formaldehyde to create methylene tetrahydrofolate. Keeping in mind that methyl group that is now attached to folate, because you got to make it active, that is deuterium depleted. That methane is not going to contain deuterium. Now, methylene tetrahydrofolate goes through its cycle where it can combine either with glycine and generate serine, and as a result, because it's passing off its methyl group, as a result, you've got tetrahydrofolate. So we're getting close to that active form, and we brought in the methyl group that is depleted of deuterium. So There's the methyl cycle that happens in the liver predominantly, where a lot of what we think of as the methylation cycle, the methyl cycle, that's where we mostly think of it happening. But this is a parallel process that's happening in the gut. So our bacteria have this very elaborate system for scrubbing incoming molecule. The hydrogen gas is coming in, carbon dioxide is there. It is building new molecules of methane, which is another way of saying methyl groups, that is depleted of deuterium. So if the system, if our deuterium regulation system that is supposed to be happening in the liver gets backed up, or that deuterium management process breaks down, or if maybe there's just an excess of deuterium coming in through probably not water, but more likely through diet, which that there's such a thing as a high deuterium and low deuterium diet. Any of those things can mess up what's going on in the liver. Well, our body's got to have access to methyl groups. It's got to be there. So, what is an option? Well, one option is to grow some excess bacteria in the gut that will do this process, that will take the hydrogen gas that's being created by other bacteria, combine it with carbon dioxide, scrub it of deuterium, make sure that we've got pure hydrogens, get it down to formaldehyde, bring that methyl group into the methylation cycle that's happening in the gut, generating glycine, serine. You've got some ammonia that's coming off in that process. The point is you're bringing these deuterium depleted methyl groups in and allowing the body access to them if they have lost access through the normal or the healthy route. Now you've got this route, which is symptomatic, but hey, guts don't care how you feel all that much. Guts care that the jobs are getting done. And the job is keep the body supplied with methane. So I think I have one more image to share with you on this. Oh yeah, this. So again, it's from the same paper, and it's like I gotta apologize with how complex these images are, but at the same time, this is happening every instant. I mean, it's just mind-boggling, really. So this is showing a couple of different paths that choline. And so, what's the best way to talk through this? Using the one kind of it's on the left side of my screen, I guess. Hopefully, it's yours. You see choline starting there and going through a set of reactions. It's going to glycine, betaine, aldehyde, and then trimethylglycine, dimethylglycine, monomethylglycine, ultimately to glycine, with these methyl groups kind of getting passed off all the way through. And those, keep in mind, those are deuterium depleted methyl groups. So ultimately, that is one kind of big long pathway for choline to go down. And the other is that choline ends up in the brain. And keeping in mind that most choline is not dietary, most of it is synthesized in the gut. And it's an extremely complicated pathway, and I won't go into all that. But choline then in the brain is either acting as a neurotransmitter as acetylcholine, or it can go enzymatically through this reaction that becomes acetaldehyde, which we all know from alcohol, ethanol breakdown. Why do we feel hangover? It's because that stuff builds up. So choline can metabolize to that and to TMA, trimethylamine, and those are deuterium depleted methyl groups. TMA can then go on to, as you see, generate methane, ammonia, these others. See all those hydrogens there, and those are all deuterium depleted hydrogen. Or now that you've gotten rid of these methyl group or these hydrogens by building all these molecules, you've got some leftover deuterium because the enzymes have scrubbed it. With the enzymes, you keep the deuterium out. So now, what can you do with the TMA that has the deuterium on it that the enzymes won't touch? Very clever. You pack it up and you send it to the liver, where it is now oxidized and it becomes TMAO. And you do a little homework, and TMAO is associated with all kinds of health problems, cardiovascular disease, and just a big and growing list of health problems that are associated with TMAO. Now, the standard idea is that, oh, well, we have to cut down on meat intake because meat is ultimately what leads to TMAO. I don't think so. The TMAO is showing up in the liver because upstream or backstream, there is a breakdown in the deuterium scrubbing that ultimately leads to the generation of these three methanes at the bottom and all that. If you can't adequately scrub the deuterium, you can't let those methyl groups get used to build the molecules that the body's trying for. So you got to send it. You got to just send it to the liver. If it's loaded with deuterium, you got to send it to the liver to get oxidized so that it can get excreted. So TMAO showing up in the blood is essentially a breakdown of the gut methylation process or the methyl cycling or recycling process where methyl groups get used to build new molecules. So uh, the growth, the excess of methane in the gut is, I think, extremely important to understand in the context of the important physiological role that methane is playing for the body's kind of the maintenance of the methylation process overall, which is happening a bazillion times a second. And so it's sort of like I think the approach of just trying to kill those bugs is is to to ignore the astounding complexity in pathways like this and all of the enzymes involved and how all of those enzymes are creating methyl groups, ultimately methane, scrubbed of deuterium so that can feed into other methylation pathways, building the organic molecules that we're trying to get, and do it without clogging it all up with deuterium. So, all right, that is my overview of kind of the methane branch of SIBO and why I think we need a more nuanced understanding of these bacteria and why they might be there and what they're up to. In future videos, and I encourage you to subscribe and like and do all that stuff that you do so that you get notified when I've got future videos. I'm going to be talking through hydrogen sulfide and giving some more thoughts around that one and many other molecules that personally I think we have sorely misunderstood in thinking of them as sort of waste products in the body when in fact they're doing something very important. So, all right, I appreciate you sitting through this. Feel free to put some comments in the comments, and I will be back soon with another version. Thanks.